Okay, we're here to finish our discussion of cardiovascular system for Part B of Chapter 18, and we want to begin with a discussion of cardiac muscle tissue, which is unique in that it's striated, branched, it has one nucleus per cell, and is connected cell to cell by special junctions that are called intercalated discs, which are combinations of desmosomes and gap junctions. Now, because of this, electrical signals, depolarizing and repolarizing waves, can spread rapidly across the myocardium, followed by a uniform contraction. The gap junctions essentially render the cardiomyocytes part of a functional syncytia, and the result then is a contraction wave that follows the electrical signal that is uniform in its direction and is essentially set by cardiomyocytes that have a depolarization rate under the influence of the parasympathetic division of the autonomic nervous system of about 75 depolarizations per minute, which is your resting heart rate. And that is set by a group of cardiomyocytes in the right atrium under the superior vena cava, known as the SA node. So this autorhythmicity is another unique feature of cardiomyocytes. The long absolute refractory period is designed to prevent the heart from going into tetany. Because of this, it's impossible to force the heart to go into anaerobic metabolism and to, as a result of the demand on the rate, beat at a rate that the cardiomyocytes are incapable of. Okay, so essentially it's a biological fail-safe against what can often happen with skeletal muscle when you overwork it to the point where it goes into lactic acid metabolism and then eventually goes into fatigue. So the heart cannot fatigue as a result of load. Now that's not to say that the heart cannot encounter a load that it can't overcome, okay, but in that case the pumping stops as a result of resistance, and we call that congestive heart failure. Similar events that take place in cardiomyocytes as do is skeletal muscle is that it's the opening of sodium channels that initiates the event. There's a reversal of membrane potential from minus 90 to 30 millivolts. The sodium channels close rapidly. The depolarizing wave runs across the entire myocardium, and it goes down into the T-tubules and results in calcium release as voltage-gated channels pop from the sarcoplasmic reticulum. That triggers the same set of events that result in skeletal muscle contraction. Calcium binds to troponin, the tropomyosin pivots out from between actin and myosin, and cross-bridging can occur. Differences include the fact that the depolarizing wave also opens slow calcium channels in the sarcolemma, and this calcium surge puts a broad shoulder on the depolarizing phase of cardiac muscle tissue. The action potential in the contractile phase lasts longer, and that permits blood to be ejected from the heart effectively. Repolarization is the result of the inactivation of calcium channels and the opening of potassium channels. The calcium gets pumped into the sarcoplasmic reticulum using ATP as an energy source, as well as being ejected extracellularly. You can see here a comparison between the changes in membrane potential and the development of tension in cardiac muscle tissue. So depolarization, the event here, is initiated again in the heart by the SA node, which is a group of rapidly, spontaneously depolarizing cardiomyocytes in the right atrium. The result is an opening of fast sodium channels and a reversal of membrane potential from about minus 90 up to plus 30 millivolts. So that's this big vertical line that you see here. And then in the plateau phase, we see calcium influx, and this keeps the cell depolarized somewhat, while there is potassium leakage into the extracellular fluid. So we are repolarizing, but we're repolarizing more slowly, because remember that calcium concentration is greater outside the cell than inside, while potassium concentration is greater inside the cell than outside. But in this case, the rate at which calcium enters and the rate at which potassium leaves are pretty close. And the result then is that we don't run the risk of throwing the cardiomyocytes into tetany. Repolarization happens when the calcium channels shut, potassium channels open wide, and the result is an efflux of potassium that brings us all the way back down to our resting voltage. And then again, the sodium-potassium pump and calcium channels are going to bring the ion concentrations back to where they were before the initiating event here. Okay. Now notice the tension that's produced in the purple wave, right? Tension is going to initiate with the sodium channels opening, okay? And it's going to peak right about where the shoulder drops off, and then it's going to drop as we have a repolarizing event taking place in earnest once the calcium channels have shut and all the potassium channels have opened. Cardiac muscle has a lot of mitochondria because it has to do a lot of work, and it depends exclusively on aerobic metabolism with very little anaerobic ability, and that's again by design. We don't want the heart to ever have to enter a state in which it has to go into anaerobic metabolism. It can switch between different fuels. It can use lactate from skeletal muscle. It can use glucose. What happens with ischemic cells is that they enter anaerobic respiration and generate lactic acid. That is going to cause the pH to drop and lead to high calcium concentration that causes mitochondrial damage and decreased ATP production. The gap junctions would close, and the result would be a fatal arrhythmia. Okay, so that's something that we don't ever want to run into. If this happens, ischemia is a pathological event, okay, and it can result from a lack of blood flow to the heart muscle itself, and that can happen as a result of a partial blockage, okay. It can happen as a result of blood loss either internally or externally. There's all sorts of events that could produce this. Now the problem, of course, is that once heart muscle tissue dies as a result of lack of oxygen supply, then it's replaced by scar tissue, and that part of the heart is always weak from that point on. The heart depolarizes and contracts independently of input from the nervous system, but it is governed by the sympathetic and parasympathetic divisions of the autonomic nervous system. Our coordinated heartbeat is the result, again, of this establishment of a functional syncytia, and the cardiac conduction system, which consists of the SA node, the AV node, the bundle of His, the right and left bundle branches, and the Purkinje fibers. And so this runs throughout the wall of the heart and promotes the movement of an electrical signal in only a certain direction. These are non-contractile cells that initiate the impulses, and then the neighboring cells are what generate the contraction. Now, the rest of the myocardium is capable of rhythmicity, but we have to be very careful. The SA node maintains the normal heart rate. If it goes out, the AV node would take over and the heart rate would fall to about 60 beats per minute. If the AV node went out, then the intrinsic depolarizing rate of the rest of the myocardium is too slow to maintain life. So it's like a race, right? The person out in front in the race is always going to be the one moving the fastest. Autorhythmic cells have an unstable resting membrane potential because of the opening of the slow sodium channels, so they continuously depolarize. At threshold, calcium channels pop, calcium rushes in, causing depolarization, and repolarization results when the calcium channels are inactivated and the potassium channels are wide open. The action potential consists of a pacemaker potential. Repolarization closes the potassium channels and opens slow sodium channels. Ion imbalance results. In depolarization, calcium channels open, calcium rushes in, a rising phase of the action potential takes place, and then in repolarization, calcium channels shut, potassium channels open, potassium rushes out. So you can see here again how this is working. We have to remember that this event happens in every cardiomyocyte, but the pacemaker cells do this more quickly than the rest of the myocardium. So the pacemaker potential is this rising area here, right? And threshold is the dotted line. So what's happening is that sodium channels are leaky, okay? And potassium channels have closed. And so sodium's drifting in at a particular rate. And then when we hit threshold, the action potential begins, okay? Depolarization from calcium influx results. And then we close the calcium channels at the peak, again, around 0 millivolts, and open the potassium channels, and then we drift back down below threshold, and we even hyperpolarize, and then we start the whole process again with the pacemaker potential. Okay, so again, it's the leaky sodium channels in this cell population, and their relative abundance that identifies the heart's wiring, the SA node, the AV node, the bundle of His, the bundle branches, and the Purkinjes, as part of the heart's conduction system. Okay, these events would also happen in the rest of the myocardium, but not quickly enough to sustain life. Cardiac pacemaker cells pass impulses across the heart in around 220 milliseconds. The SA node sends its impulse to the AV node across the walls of the atria. The AV node captures that electrical signal and pauses for about a second, little less, and that gives the ventricles time to fill with blood from the atria before we initiate a depolarizing wave at the apex that's going to work its way up the walls of the ventricles via the Purkinje fibers. The AV bundle and the right and left bundle branches rocket this impulse down to the apex of the heart through the interventricular septum, and then the Purkinje fibers bring it up the walls of the ventricles, and that's followed by a contraction that goes from the apex all the way up to the base of the heart and ejects the blood from both lower chambers. The SA node is the heart's pacemaker because it has the fastest rate of depolarization. It generates impulses about 75 times per minute, which we call our sinus rhythm, but remember that that's a result not only of the intrinsic depolarizing rate of the SA node, but the effect of the parasympathetic division of the autonomic nervous system on these cells in the SA node. So that's influence from the vagus nerve. If the vagus nerve were to be cut, this rate would kick up to about 100 times a minute. The impulse spreads across the atria, and then it's captured by the AV node, which is in the inferior interatrial septum and at the top of the interventricular septum. The impulse is delayed by about 1/10 of a second. Again, this is important. It gives the ventricles a chance to fill with blood. The reason for the extra resistance here is the smaller diameter fibers and fewer gap junctions. This lets atrial contraction finish prior to ventricular contraction and allows the ventricles to fill with blood. The rate of about 60 to 50 times per minute is the intrinsic rate of the AV node, and that can sustain life if the SA node goes out. The SA node sets what's called sinus rhythm. The AV node sets what's called nodal rhythm. The AV bundle is in the superior interventricular septum and forks into the right and left bundle branches. It's the only electrical connection between the atria and ventricles because they're not connected by gap junctions. The right and left bundle branches split towards the right and left portion of the apex of the heart. And then the Purkinje fibers bring the impulse up the walls of the ventricles. The left side has a more elaborate network than the right side of the heart. The AV bundle and subendocardial conducting network, the Purkinjes, depolarize about 30 times per minute in the absence of AV node input, and that's insufficient to maintain life. And the remaining myocardium is even slower. Ventricular contraction follows from the apex towards the atria. And again, you can see the sequence of events here. Okay, SA node begins. This spreads across the walls of the atria, is captured by the AV node. We pause for 1/10 of a second. Down the bundle of His to the right and left bundle branches to the apex. And then via the Purkinjes up the walls of the ventricles. And that's followed by a squeeze that goes from apex towards base. Okay, the same thing happens up here, right? As this depolarizing wave spreads across the walls of the atria, a contraction wave follows it, right? Depolarizing sweep first, contraction sweep second. Okay, blood goes from top to bottom, and then depolarizing sweep from apex to base, followed by contraction sweep from apex to base. So you can see here the difference in the intrinsic cardiac conduction system's depolarizing profile, right? You got time here and you've got potential here. You can see the SA node trace at the top, atrial muscle trace underneath it, and then pacemaker potential in the AV node, and then finally ventricular muscle. Okay. So again, notice in the ventricular muscle the long plateau, right? And that's to do what? That is to make sure that the myocardium cannot go into tetany. If we have problems in the conduction system, this can result in an irregular heartbeat. These are called arrhythmias. And what they do is they reduce the cardiac output of the heart. Okay. An example of this is AFib and VFib. Okay. Atrial fibrillation is where the upper chambers of the heart quiver. This is bad but not lethal because gravity actually is responsible for most of the ventricular filling in the heart. But VFib, ventricular fibrillation, is a medical emergency. The result here is that blood pumping ceases, circulation ceases, and then you're going to have brain death inside of 5 minutes if you don't get the heart going again. We use AED to treat. If you're a layman, the AED is a white suitcase. It's in a glass case embedded in the wall of wherever you work or go to school. And if you have somebody undergoing an event like this, you can remove the AED from its glass box and open the suitcase, and it will talk you through how to use it. Okay, it'll even tell you to call 911 in order to get the EMTs there so that the person doesn't succumb. A defective SA node can be the result can result in an ectopic focus, where an abnormal group of cells, a pacemaker, can form. The AV node can take over and set a nodal rhythm here, which can sustain life. An ectopic focus will result in an irregular heartbeat and not necessarily results in a lethal event. Extrasystole is premature contraction, where an ectopic focus will jump the gun before the lower chambers of the heart have completely filled and send out blood before the ventricles have reached end diastolic volume. This can result from too much caffeine or nicotine. To reach the ventricles, the impulse passes through the AV node. If the AV node is ruined, then we don't get the impulse to the ventricles, and that, again, is a medical emergency. Okay, so if you get defective AV node, we can have blockage of the signal to the lower chambers, and this can be lethal. Okay, the ventricles will beat at their intrinsic rate, but that's not rapid enough to maintain life. How do we fix this? We have to put an artificial pacemaker in the heart that is going to take over from the AV node. Okay, and so what will happen then is that the SA node will fire, the pacemaker will pick up the impulse and pass it on to the ventricles, and normal heart rate will be restored. The heart rate is modified by the autonomic nervous system, both divisions. Okay, sympathetic effect is to increase the heart rate, and parasympathetic effect is to decrease it. The cardioacceleratory center, which is part and parcel of the sympathetic effect on the heart, is going to innervate the SA and AV nodes as well as heart muscle and coronary arteries. So what's going to happen here is that under the influence of epinephrine, the SA node is going to track towards its intrinsic rate, which we know maxes at around 100. The AV node will max towards its intrinsic rate, which is around 60. Okay, and the heart muscle is going to, as a result, respond more rapidly. The coronary arteries are going to dilate in this case, and that's to bring more blood to the heart as it's working harder to pump blood around the body. The cardioinhibitory center is the vagus nerve leads to the SA and AV node, and they do the reverse. So you can see here how this operates. The vagus nerve is our parasympathetic lead. The longest cranial nerve leaves the head and neck entirely and innervates the organs in the thoracic and abdominal pelvic cavity. And you can see here how we talk to the SA and the AV node. And then over here, the cardioacceleratory center is, again, the result of neurons that run from the medulla to the lateral horn, and then we're going to hit the paravertebral ganglia, and in this case we move up a level, synapse on a postganglionic neuron, and those are the leads that are going to hit the SA and the AV node, and they're going to increase the heart rate and force of contraction, while the parasympathetics are going to bring it back towards the resting rate. Okay. An ECG is just a mechanism to measure three things about the electrical signal that moves through the heart. It measures the speed, the magnitude, and the direction of the electrical signal with respect to the electrodes that are placed at strategic locations around the heart. Now, you can do an ECG very simply. There are 3-lead ECGs, which we'll do in lab, and there are multiple lead ECGs that they do typically in a hospital or clinical setting. The main waveforms that we need to be aware of are the P wave, the QRS complex, and the T wave. Okay, you can see them here. The P wave represents atrial depolarization and has a reasonably broad shoulder. Okay, and it leads to the QRS complex, which represents two electrical events going on in the heart. One is ventricular depolarization and atrial repolarization. So the ventricular depolarization is the result, again, of an increase in the magnitude and the speed of the signal as it runs from the top to the bottom of the interventricular septum, okay, and then reverses its course going towards the Purkinje fibers, right? So the spikiness is an indication of the speed of the signal, and the height is an indication of the strength of the signal. And of course, the deflection is the indication of whether or not the signal is running away or towards the electrode in question. Several segments to be aware of. The PR interval runs from the beginning of the P wave to the beginning of the Q wave. The ST segment, the end of the S portion of the QRS complex to the beginning of the T wave, and the QT interval from the beginning of the QRS complex to the end of the T wave. Now the T wave is ventricular repolarization, and again that has a profile similar to the P wave, has a broad shoulder, the strength of the signal is relatively low, and then what follows is a repeat of the cycle. Now what's happening in each of these segments? Well, atrial depolarization leads to atrial systole, and that's going to happen about here. Okay, the QRS complex leads to ventricular systole, which is going to occur about here, as well as atrial diastole, which is occurring in the same physical location. But what you have to realize is that the signal for atrial depolarization or atrial repolarization is swamped out because of the strength of the signal going to the ventricles, so you don't really see it on the waveform. And then ventricular repolarization leads to ventricular diastole over here, right? So once ventricular diastole is complete, we start the whole process over again. The P wave fires, the atria go into systole, the QRS wave is the next waveform, next event. The atria go into diastole and the ventricles go into systole, and then ventricular repolarization, the ventricles go into diastole. Okay? And again, you can see the typical times for each of these intervals as well in a normal heart rate. You can look here and see the waveform indicated in green and the electrical signal spreading across the heart in yellow, while the event that follows the contraction isn't shown, but they do show the repolarizing wave in red. Okay, so P wave fires, SA node fires, that spreads across the walls of the atria. When it gets to the atrioventricular septum, then we start from the SA node out with atrial systole. Okay, so the atrial squeeze and the remaining ejection fraction falls from the upper to the lower chambers. And now we hold this for 1/10 of a second at the AV node, shoot it very quickly down the bundle of His and the right and left bundle branches to the apex. Okay, that's the spike here. All right, and then we're going to spread slowly up the walls of the ventricles. Okay, so we spread slowly up the walls of the ventricles. That's followed by a squeeze that goes from the apex all the way up to the base. Okay, so that's happening here. It's happening here. Okay, ventricular depolarization is complete. That's followed by a squeeze, and then repolarization, which is the T wave, spreads from apex to base, and that's followed by ventricular diastole, which is over here. Some abnormal waveforms are shown here. Okay, so in each case, there's something wrong with the typical rhythm of the heart. Now, normal rhythm is up top. Junctional rhythm, the SA node isn't working. The P waves, as a result, are absent, right? You don't see a P wave before each of these spikes, right? No P wave, no P wave, no P wave. And then the AV node takes over, right? So the AV node is going to generate a QRS complex, and you're going to get a T wave, right? But again, this is a concerning but not lethal condition, all right? And the reason is that the AV node can still generate a heart rate sufficient to support life, but it's not optimal, okay? Heart block, this is an emergency, okay? P waves don't get conducted to the ventricles. We have more P waves than QRSs, and the result then is a significant drop in cardiac output. Your effective cardiac output can drop by as much as half over under your normal rate. Okay, and this needs to be dealt with immediately. VFib. Okay, this is another immediate medical emergency, and what you got here is just a quivering set of lower chambers, and this is not an effective way to pump blood. The result can be death inside of 5 minutes. The PR interval is measured as the beginning of atrial excitation to the beginning of ventricular excitation, while the ST seg is the entire ventricular myocardium depolarized, and the QT interval is from the beginning of ventricular depolarization through ventricular repolarization. Okay, and there are certain time signatures that go with each of those intervals that are addressed in the lab podcast. Okay, so that brings this portion of Part B, Chapter 18, to a close, and we'll pick up live in class when I see you next.